Dear students, I hope you have gone through the cell transport mechanism given in the last class where we have dealt with the passive transport mechanism in detail. Here we will continue with the active transport mechanism. Okay. So, what is active transport? It is the movement of substances against the concentration gradient. It can be an electrical gradient or chemical gradient or electrochemical gradient. And this kind of transport is also known as uphill transport. See, if we can compare the passive transport to swimming along the current of water, active transport can be said as swimming against the current of water. Okay, and since here there is a movement against the gradient, there arises the need of some energy which is obtained by the breakdown of adenosine triphosphate. And this active transport occurs with the help of carrier proteins. Both ionic and non-ionic substances are transported by means of active transport. If we consider the differences between the passive transport and active transport, the prime difference is in the movement of substances. In passive transport, the movement is along the concentration gradient or down the concentration gradient, whereas in active transport it is against the concentration gradient, and hence the passive transport is known as downhill transport and active transport is known as uphill transport. In passive transport, as the movement is down the concentration gradient, no energy is required, and in active transport, since it is against the concentration gradient, energy is required. And the substances transported via passive transport are electrolytes and substances like oxygen, carbon dioxide, alcohol, water, etc. In active transport, both ionic and non-ionic substances are transported and other macromolecules are also transported. We know that in carrier mediated or facilitated diffusion also these macromolecules are transported, but the difference is that in facilitated diffusion it is down the concentration gradient and no energy is required, whereas in active transport this movement is against this concentration gradient and energy is required. Okay. This is what I have just concluded in the previous slide, the differences between facilitated diffusion and active transport. In both these mechanisms, electrolytes as well as macromolecules are being transported, but the difference is that in facilitated diffusion, the movement of the substances is along the concentration gradient, whereas in the other it is against the gradient. And in the facilitated diffusion, energy is not required, whereas in the active transport, energy is required. Okay. We have already said that in active transport mechanism, the substances are transported with the help of carrier proteins. There are two types of carrier proteins, uniport and symport or antiport. The carrier protein which transports only one substance is called a uniport, and the carrier protein which transports more than one substance can either be a symport or an antiport. When the transported molecule and the co-transported ion move in the same direction, the process is called symport. When they move in opposite directions, the process is called antiport. So symport and antiport are co-transport or coupled transport mechanisms. Okay. In antiport, one molecule will be moving into the cell, whereas the other molecule will be moving out of the cell. They move in opposite directions. And in symport, typically one ion will be moving down the electrochemical gradient and utilizing the energy of that movement. The other molecule will be moving against the concentration gradient, but the movement of both these molecules will be in the same direction. Okay, the first ion will be moving down the concentration gradient and the second ion will be moving against the concentration gradient, and the movement of both these ions will be in the same direction in symport. Okay. Active transport is of two types, primary active transport and secondary active transport. Primary active transport is the mechanism in which the energy required for the movement of substances is liberated directly from the breakdown of adenosine triphosphate. The substances like sodium, potassium, calcium, hydrogen, etc. are transported across the cell membrane by means of primary active transport. For example, sodium potassium pump is also known as sodium potassium ATPase, hydrogen pump, calcium pump. All these are examples of primary active transport mechanism. Sodium potassium pump, also known as sodium potassium ATPase, is a carrier protein which helps in the transport of sodium ions and potassium ions across the cell membrane. We know that the concentration of sodium ions is higher in the extracellular fluid when compared to intracellular fluid, and the concentration of potassium ions is higher inside the cells when compared to the extracellular fluid. And as this is an active transport mechanism, here the ions move against the concentration gradient. Okay, so this sodium potassium pump helps in the transport of sodium ions outwards the cell and the movement of potassium ions into the cell. Okay, so the sodium potassium pump is responsible for the distribution of sodium ions and potassium ions across the cell membrane and also in the development of resting membrane potential. And the structure of this pump is such that it has two protein subunits, alpha unit and a beta unit. The function of beta unit is not so known. The alpha unit has got 6 sites. 3 sites are just the receptor sites for sodium ions which faces towards the intracellular fluid, and 2 receptor sites for potassium ions which is towards the extracellular fluid, and 1 site for ATPase which is located near the sites for sodium ion. Okay. Mechanism of action of the sodium potassium pump is such that 3 sodium ions get attached to the receptor sites on the inner surface of the carrier protein and 2 potassium ions get attached to the receptor site on the outer surface of the carrier protein. When these ions get attached to their respective receptor sites, the enzyme ATPase will be activated, and the ATPase causes the breakdown of ATP into ADP and the release of one high energy phosphate occurs. This liberation of energy causes some kind of conformational change in the structure of the carrier protein, resulting in the interchange of the surface of the carrier protein. That is, the inner surface of the carrier protein faces the outer side of the cell after this conformational change, and the outer surface of the protein molecule now faces the inner side of the cell. So now in this interchange position, the dissociation and release of the ions take place. So what happens? The sodium ions will be released into the extracellular fluid and the potassium ions will be released into the intracellular fluid. Okay, so the sodium ions from inside the cell are now transported across the membrane outside the cell, and the potassium ions from outside the cell are now transported into the cell. So this is how the transport of sodium ions and potassium ions happen with the help of sodium potassium pump. This means that for every 2 potassium ions that are pumped into the cell, 3 sodium ions are pumped outside the cell. So there is a net loss of 1 positive charge for this cell. Okay, so when this sodium potassium pump continues to function, there develops a negative potential inside the cell. We know that the resting membrane potential of a cell is in the range of -90 to -70 millivolt depending on the type of cell. Okay, so anyways, the resting membrane potential is a negative potential, And the sodium potassium pump helps in the development of this resting membrane potential. Okay. Next is secondary active transport. In this mechanism, the transport of molecules occur across the cell membrane utilizing energy in other forms than ATP. This energy comes from the electrochemical gradient created by the ions pumped out of the cell. So mostly it is associated with the sodium ions. We know that by the primary active transport in the sodium potassium pump, the sodium ions are pumped out of the cell and the concentration of sodium ions outside the cell is much higher when compared to the intracellular fluid. So there is an electrochemical gradient with respect to sodium ions. So this gradient becomes energy as the excess sodium ion constantly try to diffuse to the interior of the cell. Okay, so when the sodium ion is transported by means of a carrier protein, another substance is also transported by the same carrier protein. It can happen either in the same direction or in the opposite direction. If it's in the same direction, it is called a co-transport, and if it is in the opposite direction, it is called a counter-transport. So in sodium co-transport, along with the transport of sodium ion, another substance is also carried by the same carrier protein in the same direction. Substances usually carried along with sodium are glucose, amino acids, chlorides, etc. So here, as we have seen, because of the sodium potassium pump in the primary active mechanism, the concentration of sodium ions is higher in the extracellular fluid, creating an electrochemical gradient. And because of this gradient, the sodium ions continuously try to move back into the cell. So the energy released by the movement of sodium ion is utilized for the movement of the substance that is transported by the same carrier protein. Here, so ATP is not directly utilized and hence it is called a secondary active transport. Okay. And in sodium co-transport, the carrier protein has two receptor sites on the exterior of this carrier protein that is facing the extracellular fluid. One side is for the transport of sodium ion, for binding the sodium ion, sodium receptor site, and the another site is for the attachment of the substance that is carried along with sodium ion, maybe glucose, amino acid, etc. So in case of sodium co-transport of glucose, one sodium ion gets attached to the receptor site of the sodium ion in the exterior of the carrier protein, and one glucose molecule gets attached to the respective receptor site for it. When both these substances get attached to the carrier protein, some conformational change occur so that the sodium ion and glucose ion will be released into the cell. This sodium co-transport occurs in the intestine while the absorption of glucose, and in the renal tubules while the reabsorption of glucose from the filtrate. Okay. In sodium counter transport, the substances are transported in exchange for sodium ions by the carrier protein, which is an antiport. Here the ions are transported in the opposite direction. Okay. And the examples are sodium calcium counter transport, which is present in almost all the cells. Then sodium hydrogen counter transport. Here the hydrogen ions are exchanged for the sodium ions seen in renal tubular cells. Then sodium magnesium counter transport, sodium potassium counter transport, calcium potassium counter transport. Likewise, many examples are there. Apart from the primary and secondary active transport mechanisms, there are some special types of active transport systems where the substances are transported through the cell membrane as vesicles. Hence, such mechanisms are generally called as vesicular transport. Major types of vesicular transport are endocytosis and exocytosis. Endocytosis is the mechanism where the macromolecules enter the cell. Since they are larger in size, they cannot pass through the cell membrane by active transport or passive transport. Such substances are transported into the cell by means of endocytosis. There are three types of endocytosis: pinocytosis, phagocytosis, and receptor-mediated endocytosis. Next is exocytosis, where the substances are expelled out of the cell in forms of vesicles. And this is the reverse of endocytosis. Actually, this pinocytosis, phagocytosis, etc., you know well. You have learned in your smaller classes. So I'm not going to explain in detail. Okay. So the process where the macromolecules are taken into the cells in the form of fluid droplets is called pinocytosis. It is otherwise known as cell drinking. So the macromolecules in the form of droplets of fluid bind to the outer surface of the cell membrane, and the cell membrane evaginates around the droplets, and these droplets are engulfed by the cell membrane, and the engulfed droplets are converted into vesicles and vacuoles called endosomes, and these endosomes travel into the interior of the cell. Then the primary lysosome attaches to this endosome and form the secondary lysosome, and once the secondary lysosome is formed, the hydrolytic enzyme present in these secondary lysosomes are released, resulting in the digestion and degradation of the endosomal contents. Okay. Phagocytosis is a process where substances larger than the macromolecules like larger bacteria, larger antigens, etc. are engulfed into the cell. Hence it is also known as cell eating. And when such larger foreign bodies enter the body, the phagocytic cell sends a cytoplasmic extension called pseudopodium around this foreign body, and these particles are engulfed and converted into endosome like vacuoles. And in case of phagocytosis, such endosomes are called phagosomes. And these phagosomes travel into the interior of the cell, where the primary lysosome fuses with it and form the secondary lysosome, and the hydrolytic enzymes which are activated results in the digestion and degradation of the contents in the phagosomes. Only a few cells in the body have this phagocytic activity like monocytes, neutrophils and macrophages, and among them macrophages are the largest phagocytic cells. Next is receptor-mediated endocytosis. This is the transport of macromolecules with the help of a receptor protein. On the surface of the cell membrane, there are pits which are coated with receptor protein like clathrin, caveolin, etc. So these pits together with this receptor protein is called receptor coated pit, and they are involved in the receptor-mediated endocytosis. This receptor-mediated endocytosis is induced by substances like ligands. So these ligands, when comes near the cell, binds to the receptor in the coated pits, form a complex called ligand receptor complex, and this ligand receptor complex gets collected in the pit, in the coated pit, and then it gets detached from the membrane, becomes a coated vesicle and form endosome. Then it travels into the interior of the cell. Primary lysosome gets fused with this endosome to form the secondary lysosome, and the hydrolytic enzymes inside this lysosome gets activated, resulting in the release of these ligands in the cytoplasm, into the cytoplasm. And this receptor is again moving to the cell membrane to a new pit. The macromolecules which are transported by this receptor-mediated endocytosis are many hormones like growth hormone, thyroid stimulating hormone, luteinizing hormones, etc. The lipids, the growth factors, toxins and bacteria, viruses, antibodies like IgE, IgG, maternal IgG, etc. Next is receptor-mediated endocytosis. This type of transport of macromolecules occur with the help of receptor proteins. On the surface of the cell membrane, there are some pits which contain the proteins like clathrin, cabulin, etc. So these pits together with the receptor protein is called receptor-coated pit. And these receptor-coated pits are involved in the receptor-mediated endocytosis. This type of transport is induced by the substances like ligands, and these ligands, when come near the cells, they bind to the receptors in the pits to form ligand receptor complex. The ligand receptor complex, when gets aggregated, the pit is detached from the cell membrane, becomes a coated vesicle, forms the endosome and moves into the interior of the cell. Rest is the same. Primary lysosome fuses with this endosome to form secondary lysosome. The hydrolytic enzymes get activated, resulting in the release of the ligands into the cytoplasm, and the receptor moves back to a new pit in the cell membrane. The macromolecules which are transported by this mechanism are hormones like growth hormone, thyroid stimulating hormone, luteinizing hormone, prolactin, insulin, glucagon, etc. Lipids, growth factors, toxins and bacteria, viruses, antibodies, IgE, IgG, maternal IgG, etc. Exocytosis is the process where the substances are expelled from the cell without passing through the cell membrane, and it is the reverse of endocytosis. The release of secretory substances from the cells occur by means of exocytosis. These secretory substances are stored in the form of secretory vesicles, and when required, these vesicles fuses with the cell membrane and the contents of the vesicles are released out of the cell. Calcium ions are having an important role in the release of these secretory substances. In applied physiology, the abnormality of sodium-potassium pump can result in many pathologies, like a decrease in the sodium-potassium pump in myocardium can cause cardiac failure, whereas an increase in the sodium-potassium pumps in renal tubules can cause hypertension because of the increased tubular reabsorption of sodium ions. Thus, the cell transport mechanism has been completed. Now, dear students, I'd like to give a very small assignment, nothing strainful. Just you read the topic transcytosis and also molecular motors and make a very short note on molecular motors. Okay, thank you.